This is an overview of an article titled Cholesteryl Ester Transfer Protein, or CETP, Concentration, a genome-wide association study followed by Mendelian randomization on coronary artery disease, published in the May 2018 issue of Circulation, Genomic and Precision Medicine. Cholesteryl ester transfer protein, or CETP, facilitates the net flux of cholesterol esters from high-density lipoproteins, or HDL, towards very low-density lipoproteins, or VLDL, coupled to a net flux of triglycerides from VLDL to HDL. CETP contributes to an atherogenic lipoprotein profile, that is, a high LDL cholesterol to HDL cholesterol ratio. Therefore, inhibition of CETP has long been regarded as a potential therapeutic strategy to attenuate dyslipidemia and ultimately prevent the development of cardiovascular disease, or CVD. The 4 clinical trials that have studied the effects of pharmacological CETP inhibition on CVD risk reduction have been largely disappointing, and only the most recent, the REVEAL trial with anacetrapib, met its primary endpoint. Intriguingly, anacetrapib showed the largest reduction in the LDL cholesterol concentration compared to the 3 previously tested CETP inhibitors. The genetic basis of serum CETP, which is derived from hepatic macrophages, has yet to be determined. Therefore, the authors undertook a genome-wide association study, or GWAS, to identify genetic variants associated with CETP levels. In addition, they wanted to determine whether the genetic variants associated with CETP concentrations were also associated with serum lipid levels and CVD risk by utilizing Mendelian randomization. The premise of a Mendelian randomization study is that if a biomarker is causal for a disease, then genetic variants that influence the levels of the biomarker should result in a higher risk of the disease. With respect to CETP and CVD, there are multiple potential mediators of a causal effect, including LDL cholesterol and HDL cholesterol. In the paper under discussion, the authors performed a GWAS on serum CETP concentration using a discovery cohort with 4,248 individuals and a separate replication cohort with 1,458 individuals from the Netherlands Epidemiology of Obesity, or NEO, study. Then, they used the GWAS-identified single nucleotide polymorphisms, or SNPs, in Mendelian randomization analyses on serum lipid concentrations in the NEO study population and the Global Lipids Genetics Consortium, and on coronary artery disease, or CAD, using publicly available data from the Cardiogram plus C4D consortium. The figure on the left shows the results of the GWAS analysis, with negative log p-value on the y-axis and chromosomes on the x-axis. After conditioning on the lead SNPs, 3 independent variants reached genome-wide significance with a conditioned p-value of less than 5 × 10 to the negative 8th. The 3 genome-wide significant variants all mapped to the CETP locus, resulted in increases in serum CETP levels, and replicated in the replication analysis. The figure on the right shows the regional association plot for the top signals at the CETP locus with the lead SNP, RS247616, shown in purple. These graphs show the results from the Mendelian randomization analyses on CAD risk and serum lipid and apolipoprotein B, or ApoB, concentrations. In the top left panel, you can see that for each 1 microgram per milliliter increase in genetically determined serum CETP concentration, the odds ratio for CAD risk was 1.08. The odds ratios for CAD risk for the 3 individual SNPs are also shown, with 2 of them achieving statistical significance. The other panels show the results on serum lipids, a 1 microgram per milliliter increase in genetically determined serum CETP concentration was associated with decreased total cholesterol concentration and HDL cholesterol concentration, while it was associated with increased LDL cholesterol concentration and ApoB concentration. This table shows the results of expression quantitative trait locus, or eQTL, analyses for the 3 lead SNPs identified in the GWAS. The SNP that was most strongly associated with serum CETP concentration in the GWAS, RS247616, was identified as an eQTL for the CETP gene in several tissues. In summary, first, this study confirms that variants in the CETP locus are the major genetic determinants of serum CETP levels. The top 3 independent SNPs explain 16.4% of the total variation in CETP concentrations. Second, genetically determined variation in circulating CETP associates with a stepwise substantial decrease in the HDL cholesterol concentration, a moderate increase in LDL cholesterol and ApoB concentrations, and a concordant 8% increase in CAD risk. Finally, together, these findings, along with previous Mendelian randomization studies of circulating HDL cholesterol and LDL cholesterol concentrations, suggest that the causal association between CETP concentration and CAD risk might be explained by effects on LDL cholesterol concentration.